Welcome back to the International Virtual Conference 2020. I would like to call Mrs. Rita Ma'am, Assistant Professor, Department of Chemistry, to introduce the guest speaker of the session 3, Dr. Tyagarajan sir. Over to Rita Ma'am.
Thank you, Bharati. Good morning to one and all present here. On behalf of Anai Violet Arts and Science College, Department of Chemistry, Biochemistry and Physics, it is indeed a great pleasure and privilege to introduce our eminent invited speaker of this session, Dr. Tyagarajan. We are honored and would like to express our gratitude for accepting our invitation to deliver a lecture for this session and gracing it with august presence, sir. Dr. Tyagarajan has completed his doctoral degree in University of Madras and currently he is working as assistant professor and coordinator in Northern Border University, Kingdom of Saudi Arabia. He has 25 years of experience and has more than 20 years of research experience in epidemiological and diagnostic studies of infectious diseases, especially of viral origin. Awards or titles conferred by Dr. Tyagarajan. He has received Anand Rao Memorial Award of Indian Association of Applied Microbiologists in the year 2009. And also he has received Professor S. Anandan Endowment Award of International Association of Medical and Pharmaceutical Virologists in the year 2011. He was also nominated as Science Communicator by Indian Science Congress Association in the year 2011. He got a fellowship title, Fellow of Indian Association of Biomedical Scientists in the year 2011. Furthermore, he served as a member of Board of Studies for designing microbiological curriculum for University of Madras and other autonomous colleges. With this introductory note, I would like to call upon our prestigious invited speaker to take over the session. Dear participants, please join us in providing a hearty welcome to our resource person of this online webinar session. We welcome you, sir. One announcement. Participant can post your questions in chat box during the session and fill the feedback at the end of the session. Now over to Dr. Tyagarajan.
Thank you, madam, for introducing me. It's my privilege to participate in this international virtual conference. I'm happy that our people have taken effort and enthusiasm in organizing this conference for the benefit of the students. This is a platform to share our knowledge, share the information, what we have acquired through various media. So in this virtual conference, I would like to present some of the scientific information which I have acquired through going through various research articles pertaining to the fields of microbiology and biochemistry. This invited talk is going to be about Helicobacter pylori induced autoimmune thyroiditis. Many would wonder, what is the relationship between a bacteria Helicobacter pylori and the autoimmune disease thyroiditis? Yes, there is a connection between these two. This is what is the surprising information which attracted me to learn about this and bring out an article out of it. So I thank the organizer for inviting me to participate in this conference. Let me share the slide. Ladies, are you able to see the slide?
Sir, no, sir. You have to share your screen, sir.
Yes. Should we wait for some more participant to come?
Sir, you can proceed, sir. You can click that slideshow and you can proceed, sir.
Yeah. So this lecture is about Helicobacter pylori induced autoimmune thyroiditis and insight into its pathogenesis. Before I get into my talk, let me introduce the institution where I belong to. So this is what is our my institution where I belong to, where I am working currently, the Northern Border University, located in Arar, Saudi Arabia. This university is a government university controlled by Ministry of Education. It was instituted in the year 2007, relatively a new university, one among the more than 50 universities of government universities of Saudi Arabia. And this university is located in Arar, the capital of northern province of Saudi Arabia. And this university offers an array of courses, including medicine, engineering, pharmacy, nursing, computer education, business administration, community science, medical sciences, and number of courses are offered. Almost 16 colleges are affiliated to this university and 13 deanships are there to manage this institution. And this college, this university is located, has 3 branches in addition to its main campus in Arar, like Turayf, Wagila, and Rafah. So this is the entrance of our institute and administrative buildings, and this is the Faculty of Applied Medical Sciences where I am currently working with. I am thankful to my institution for giving an opportunity to work under this prestigious institute, where I could see many eminent professors and scientists working with more of enthusiasm and interest to contribute for the scientific advancement. Okay. So let me take you to the main part of my lecture, that is Helicobacter pylori induced autoimmune thyroiditis, an insight into its pathogenesis. You know, this lecture, as I mentioned before, it is a combination of microbiology, biochemistry, and an amalgamation of immunology. As I mentioned before, this bacteria, the Helicobacter pylori, is basically a gastric pathogen which causes infection in the stomach. So most of us believe that this causes infection only in the stomach and it is no way connected with other diseases. But it is, interestingly, is capable of causing variety of diseases, not only the disease in the gastric system, but also in various other anatomical parts of the body. That is what we are going to see in detail in the lecture. This lecture comprises 3 components. The first component is about the biochemistry of thyroid. And the second component is about microbiology concerning the bacteria Helicobacter pylori. And third component is about the link between these two. What matters, the link between these two, the bacteria and thyroiditis. Okay, let me start with the first part of my lecture, thyroiditis and its autoimmune origin. Thyroid disease, you know, generally we call it as thyroid disease. What is thyroid disease? Thyroid disease is nothing but a disease associated with the thyroid, which is a very, very important endocrine system. And this thyroid disease usually manifests as thyroiditis. As the name indicates, thyroiditis, it refers to inflammation. So it is a disorder of thyroid which has multiple etiology. We cannot say a particular cause or particular reason for thyroid disease. There are many different causes. It has multiple etiology. In general, thyroiditis refers to inflammation of the thyroid gland. Inflammation is enlargement which is brought about by number of immunological mechanisms. And what happens? Because of this inflammation of thyroid gland, there occurs altered function of the gland. The function of thyroid gland is altered in such a way that sometimes there is over secretion of the thyroid hormones, or sometimes under secretion, or sometimes there is nil secretion. So whatever it may be, the abnormal functioning of thyroid gland affects many of our metabolic pathways, because endocrine system is very important, particularly thyroid based endocrine system is very important in controlling number of metabolic pathways. So any malfunctioning, dysfunctioning of thyroid gland will affect the entire functioning of the body. So the diseases concerning with the thyroid are categorized broadly into two conditions. There are two conditions of thyroid disease, which are further classified into 5 types based on the causes that are responsible and the clinical features. In this slide, you can see the common types of thyroid diseases. You can find two important conditions, two major conditions of thyroiditis, called acute thyroiditis and thyroid dysfunction. Acute thyroiditis, acute means it occurs with a sudden onset, which has a relatively durable course. It can be recovered. It can be recovered, or sometimes it can progress to a chronic condition. But in most cases, it is recovered. The acute thyroiditis can be either infectious or a granulomatous type of condition. Infectious condition refers to the infection caused by some microbial pathogens in the thyroid gland that can lead to alteration of the function of the thyroid gland. Granulomatous region condition is purely a immunological mechanism that causes alteration in function of thyroiditis. It is an acute condition which affects mostly people, the infants who are malnourished, sometimes the old age people. Most importantly, the immunocompromised patients are the main targets of this condition, the infectious disease, infectious thyroiditis. The granulomatous thyroiditis usually affects middle-aged women. The second condition, called thyroid dysfunction, can be further categorized into lymphocytic, Hashimoto or stroma, or lymphomatosa, and riddle thyroid dysfunction. So in this case, in all these cases, you can see there occurs total failure of function of thyroid. So thyroid gland stops its function because of malfunctioning caused by variety of causes. If you observe the people who are affected, mostly the children are affected by thyroid dysfunction, and women who are over 40 years of age, and in some cases, adults and elderly patients are affected. In most of these cases, you see, young children and women are more susceptible to thyroid dysfunction or acute thyroiditis. So the common clinical features include pharyngitis, tonsillitis, and you will have generalized sepsis. The symptoms include sore throat. It starts as a common sore throat. There will be painful throat, tenderness of thyroid region. Patients will have on and off fever. And most important feature of this thyroiditis is the goiter formation. So goiter is the enlarged thyroid gland, resulting in the repeated inflammation, repeated immunological reactions and inflammation. So the condition called autoimmune thyroid disease, it is a collection of different diseases concerning with thyroid. It is one of the most frequently reported autoimmune diseases. One must know what is an autoimmune disease. For those who need basic information on autoimmune disease, I would say autoimmune disease is a condition where our immune system acts against our own body tissues and cells or organs. Immune system is meant for defending the body against deleterious agents, foreign agents. But sometimes it so happens that our immune system gets overreacted, and it acts on our own body tissues. So this is a undesirable condition. It can affect normal functioning of the body. So it so happens in the case of the autoimmune disease can be either hematological or organ specific or sometimes systemic. Some autoimmune diseases are pertaining to particular organ. Maybe it affects only joints, bones, or it can affect a particular organ like thyroid or pancreas or stomach. Likewise, this autoimmune thyroid disease is a condition where our immune system acts against the thyroid gland. It is a very important major condition that predisposes to thyroid dysfunction. This autoimmune disease is a collection of different diseases, which includes Hashimoto's thyroiditis, Graves' disease, thyroid-associated orbitopathy, postpartum thyroiditis, and atrophic autoimmune hypothyroidism. See, if you see all these autoimmune thyroid diseases, the most important autoimmune thyroid diseases are Hashimoto's thyroiditis and atrophic autoimmune hypothyroidism. These two are considered the most important autoimmune thyroiditis, conditions leading to autoimmune thyroiditis. And the difference between these two is Hashimoto's thyroiditis is often associated with goiter formation, enlargement of thyroid, whereas atrophic autoimmune hypothyroidism is a condition which is where there is no goiter. So these two are most frequent and most common autoimmune thyroiditis is Hashimoto's thyroiditis. So what I'm going to refer in this lecture, the autoimmune thyroiditis is fully concerning with Hashimoto's thyroiditis. Okay, pathogenesis of autoimmune thyroiditis. How does it occur? You know, the Hashimoto's thyroiditis, which is what is commonly known as autoimmune thyroiditis, which is characterized by enlargement of thyroid glands. The thyroid glands usually enlarges. It enlarges to maybe 10 times or 20 times. So sometimes, rarely you would see people having large thyroid gland which is hanging from the neck. It was first described by Hakura Hashimoto in the year 1912, who referred it as chronic lymphocytic thyroiditis. What did he do? He examined the thyroid gland, histotechnological studies he did, and he found that there occurred more infiltration of lymphocytes. So he believed that it is because of the lymphocytes the enlargement of thyroid occurs and it causes thyroid dysfunction. It is basically a chronic inflammation. The inflammation is not acute, it is a chronic inflammation. It occurs for a long period, prolonged period. Unless it is attempted seriously, it will not cure. It is mainly because of infiltration of lymphocytes, different types of lymphocytes, B lymphocytes, T lymphocytes, even natural killer cells. All these cells infiltrate and carry out continuous immunological reactions resulting in inflammation and further enlargement of thyroid gland. The most important feature of this autoimmune thyroiditis is the Hashimoto's thyroiditis. There occurs the presence of circulating autoantibodies which are directed against thyroid antigens. You know, thyroid gland is a complete discrete structure which has many different components, and these components in terms of immunology we mention as thyroid antigens. So antibodies are produced against the thyroid antigens. The most common thyroid autoantibodies are thyroglobulin antibodies, thyroid peroxidase antibodies, thyrotrophin receptor antibodies. These are the most common types of autoantibodies produced during the autoimmune thyroiditis. The problem is these autoantibodies interfere with the function of thyroid, so that thyroid secretion is affected. Sometimes it is not secreted, or sometimes it is secreted in excess. Both the conditions lead to, both are pathological. Okay, what is the etiology of autoimmune thyroiditis? How does it occur? What is the cause? Several causes have been proposed, of which two are more important, called genetic factors and environmental factors. So how about the genetic predisposition? Genetic predisposition, I would say predisposition means it is already determined by the genes, by the chromosomes, what you inherit from your parents. So there occurs inheritance of certain susceptible genes among the family. So if the parents have susceptible genes which predisposes the autoimmune thyroiditis, the children also would have. There is every chance. And you can see this is evident in the case of twins. So twins who are born at the same time from mother, they both of them, if one gets thyroiditis, you can find the other also gets immediately or at the simultaneously. It is because of the inheritance of genes. So these genes, what happens? These genes, when they are expressed, there occurs loss of immunological autotolerance. What is autotolerance? So I need to explain briefly about this autotolerance. Autotolerance is nothing but the mechanism that causes autoimmunity. Autoimmunity. What is autoimmunity? There is a difference between autoimmunity and autoimmune diseases. So autoimmunity is a condition where our body ensures a homeostasis. Homeostasis. What is homeostasis? Homeostasis is a kind of understanding between the immune system and our body's tissues. You know, every body cells, every tissue, every cell of our body is provided with an array of chemicals. These chemicals, in terms of immunology, from the perspective of immunology, you can say they are called as autoantigens or self-antigens. You know, as long as these antigens are in your body, you call it as self-antigen. When it is received from other body, it is called foreign antigen. For example, I would say the blood group antigens, blood groups, you know, A, B, O. Besides, you have RH antigens, D antigens. So these antigens, these blood groups are called, the antigens are found on the surface of your RBC, which we refer as self-antigen, against which there is no immunological reaction. Whereas when you receive a different antigen of same blood, different blood group from other person, immediately the immunological reactions are mounted. So as long as any antigen or any chemical which is found in your body, which is recognized by your immune system, it is called self-antigen. So immune mechanisms are not mounted because of a condition called autotolerance. This is because of some immune mechanisms which are developed during your embryonic life. You know, when embryo is formed, when alongside the organogenesis, the organs are formed, the immune system is also formed. Both of them are encountering each other. During the initial phase of your fetal development, clones of cells are produced, which are autoreactive in nature. Means the immune system in the beginning, when the fetus is formed, immune system produces antibodies and cells, not exactly antibodies, the cells, which can react with our body's antigens. And in due course, they are all removed. They are all cleared, and which are referred to as forbidden clones. This is an immune mechanism. So these forbidden clones are removed from the body so that immunity or immune system does not recognize your body's tissues or body's antigens, which is what is called as autotolerance. Okay. So this autotolerance is affected because of the expression of certain genes. So this predisposition of genetic factors is controlled is because of some haplotypic genes in our body. Some haplotypic genes are there. What are those? These haplotypic genes include HLA-DQ2, HLA-DQ8, and CTLA4. These are the most important. Besides these, you have many other genes. These genes, they are the ones which are responsible for mounting immunological reactions against self-antigens. Whatever the tissues and the organs we have, all of them have self-antigens. Against these antigens, the immunological reactions are mounted if these genes are expressed. These genes are expressed. So it so happens in the case of people having thyroiditis, these genes are expressed. They are not controlled. They are not recessive. They are in an expressive form. So that there occurs autoimmune thyroiditis. So resulting in the production of the clones of cells, autoreactive clones of cells and antibodies. In addition to your genetic factor, not that everyone would get thyroiditis because of the inheritance of genes, but also environmental triggers can also lead to thyroiditis, which includes smoking, some medications, irritation, irradiation, stress, and most importantly, the iodine intake. The iodine intake is more associated with the thyroiditis. So wherever you can see in the population where they take more of iodine, you can find the higher prevalence of thyroiditis. Whereas you can find limitation or less prevalence of thyroiditis in such areas, in such cases where the iodine deficiency occurs. Some populations have iodine deficiency where you can hardly see the thyroiditis. But in addition, you may have transient thyroiditis in the case of pregnancy. Some pollutants can also cause induce thyroiditis. And most importantly, the infections. We now and then encounter different infections by bacteria, viruses, fungi, yeast, and many parasites. They cause infections. These infections are considered to be critical environmental influences for the development of autoimmune thyroiditis. Why? It so happens some bacteria, some viruses, not all of them, they have some antigenic structures. Antigens means chemicals which are found on the surface or are secreted from the pathogens, bacteria or viruses. And these antigenic substances, they mimic the antigenic components of thyroid membrane. Thyroid gland is made up of membrane. This membrane has some structures, some chemical structures, which are mimicked by some antigenic components possessed by certain bacteria and viruses. Mimicry, the meaning of mimicry. Mimicry means resemblance or doing something which is similar to something. So because of the similarity, structural similarity between the antigens of microbes and the thyroid membrane or thyroid gland, there occurs an immune reaction against the pathogens which also tend to react with the thyroid gland. This is how the autoimmune thyroiditis develops. So the infections play a very, very critical role in development of autoimmune thyroiditis. Okay, how about the prevalence and significance of autoimmune thyroiditis? Why it is given so much importance? While so many diseases are there, why autoimmune thyroiditis is given so much importance? Globally, the Hashimoto's thyroiditis or autoimmune thyroiditis is referred as the most common type of thyroid disease. Although too many thyroid diseases are there, the Hashimoto's thyroiditis, that is autoimmune thyroiditis, is the most common type. Not only in children, but also in the adolescents. So this condition is frequently implicated with hypothyroidism. So there occurs less production of thyroid. This is a very alarming condition which can clinically manifest either with goiter formation or without goiter formation. Usually this disease occurs at the age of 3 and it progresses slowly. Symptoms are not seen. At a very early age, at the age of 3, the disease onset and clinical characteristics are seen usually after 6 years. And the symptoms peak around the age of 10 and 11 years. After that, you can view the severity of the disease. You can experience the severity of the disease. It is estimated that the people who are at the age of 40 to 60 years are commonly affected, and the incidence rate, if you see the incidence rate, it is 3.5% among the women and 0.8% for men. Per every 1,000 population of thyroiditis affected people, if you say 3.5% of people are The Hashimoto's thyroiditis has been demonstrated to be closely associated with the gastric diseases. So who are all the people more susceptible? Who are all the people who can more predisposed to thyroiditis? If you see, those who have gastric diseases, the diseases associated with the stomach. So it has a high prevalence in patients with gastric disorders, and the autoimmune gastritis. There is another disease called autoimmune gastritis where antibodies are produced against our own stomach system, some components. So those who have autoimmune disease or autoimmune gastritis, they have nearly 10 to 40% chance of developing autoimmune thyroiditis. Okay, let us move on to the second component, that is the Helicobacter pylori. This bacteria is considered as a precarious bacteria, dangerous. It is a bacteria which is considered as a dangerous bacteria among all the bacteria. Why? Because this bacteria is a peculiar bacteria which we refer as gastric bacteria. Normally bacteria, when we refer, depending on the infection, we refer them as the enteric bacteria or septicemia causing bacteria or respiratory bacteria, urinogenital infection causing bacteria. Different regions, based on the regions and the diseases, the infection the bacteria causes, we refer them as respiratory, urinary, or gastric or digestive. But this is the bacteria which is called as gastric bacteria. Gastric bacteria, I would say, gastric bacteria are very rare, and most predominant gastric bacteria is the Helicobacter pylori. Why? The gastric system, the stomach is considered to be a very, very important system of our body, and it is a unique system of our body because it possesses gastric juice. The gastric juice is made up of hydrochloric acid and multiple enzymes which all make the environment an acidic environment. The pH of this stomach usually ranges between 2 to 3, acidic pH. At this pH, there is no possibility of any bacteria to survive. You know that? Bacteria usually prefer a pH of 7 or above. Alkaline pH, slightly alkaline pH they prefer, or slightly acidic pH. But very low acidic pH is detrimental to many bacteria. But this bacteria can sustain the acidic environment and survive in that condition. That way it is peculiar. And besides some bacteria like Mycobacterium tuberculosis, this is the only successful bacteria that can survive in the stomach. This bacteria is a gram-negative bacteria. Those who know microbiology, they would know what is gram-negative. Bacteria are classified into gram-positive and gram-negative based on their staining characteristics. It is a spiral-shaped bacteria, motile in nature. It shows extraordinary motility, movement, which it has lophotrichous flagella found at one end of the bacterium, and it shows corkscrew movement. Corkscrew, how it is screwing and removing the cork. Likewise, it has a movement so that it is capable of penetrating any tough tissue, and it is a microaerophilic bacterium, so it can live in low oxygen condition. So it is known for colonizing and infecting the gastric mucosa, especially the mucosal system of gastric system. Mucosal lining of gastric system is usually colonized by this bacteria. This was first described by two scientists, Marshall and Warren from Australia in the year 1984. This bacteria is considered as most important pathogen because it has a wide-reaching chronic infection. It causes wide-reaching chronic infection. So it is widespread. So the importance of this bacteria was not realized in the beginning when it was discovered. It was not realized. People thought it is one among the normal bacteria that infects the digestive tract. Some people thought, okay, it infects only the stomach and causes some gastritis and nothing more about it. But later it was found that it causes complication not only in the stomach but also in the entire body. In multiple organs it can cause complications. The infection can cause complications. So after understanding the importance of it, it was only in the year 2005 the discoverers were given Nobel Prize. So after many years of understanding the significance of this disease, this bacteria and the associated diseases, the discoverers were given Nobel Prize. Let's see about the pathogenicity of this Helicobacter pylori. As I mentioned before, stomach is the major reservoir of this bacteria. It can be transmitted by some routes like fecal-oral route, oral-oral route, and gastro-oral route. So fecal-oral route is the most common mode of transmission. So it is transmitted through contaminated food, contaminated water, fecal contaminated water, because it is found in the digestive tract. The bacteria shed in the feces, the stool. So the bacteria is usually associated with the disease of stomach, causes many obstructions, many ulcerations, many reflux changes, and some cases, in serious cases, it can cause cancer of the stomach. That is called gastric cancer. Why? How? It possesses extraordinary virulence characteristics. So what is virulence? The ability of causing infection, power. It is a strong pathogen that it can cause infection, chronic infection, and evade host defenses. Most important point. So the pathogen not only causes infection, but also defends the defense mechanism. Means it can neglect or it can overcome the immunity developed by the individual. So whatever the immunity we develop, it becomes ineffective unless it is given care, unless the treatment is given proper care. So it can overcome the immune reactions. This bacteria possesses some of the many virulence factors. It possesses many virulence factors. I would like to say some of the important virulence factors that aids this bacteria to become a successful pathogen in stomach. So it contains some virulence factors like urease enzyme. This apart from the pathogens which infect urinary tract, this bacteria which is residing in the stomach is capable of secreting an enzyme called urease. And this urease is the one that helps the bacteria to survive in the stomach. How? This bacteria causes a buffering effect in the stomach by converting the urea into carbon dioxide and ammonia. So that a buffering effect is caused in the stomach. In that buffering effect, with that buffering effect, the bacteria is able to survive. And it has an adhesin-like proteins. Adhesin means the proteins which are helpful, which are helping for attachment of the bacteria on the epithelial surface of the gastric mucosa. And it has a very, very important antigen called cytotoxic protein. Cytotoxic protein A or CAGA. It causes or it induces an antigen or cancer. That is why it is called as a direct mutagen. It is a mutagen. This protein is a mutagen. Like any other chemical, this can cause mutation, mutagenesis in the cell where it is deposited. So this protein is the one that is responsible for peptic ulceration and gastric cancer. So this protein secreted by Helicobacter pylori is a major culprit of causing gastric cancer, in some cases laryngopharyngeal carcinoma. So not only in the stomach, but also in the larynx, in the pharynx, it can cause carcinoma, the epithelial cancer. In addition, it also secretes a vacuolating toxin. So besides possessing this protein, mutagenic protein, KAGE, it also secretes a toxin called vacuolating toxin, which plays a very, very important role of suppressing the immune system, particularly T cell proliferation. You know, the T cells, CD4 cells, they are inhibited by this vacuolating antigen. That thereby the bacteria is able to evade the immune system. That is what I said. So the bacteria produces some enzymes and proteins to survive in the stomach and produces some antigens to evade the immune system or defend the immune system so that it becomes more successful in living in the stomach. So it can induce both types of immunity, like humoral mediated immunity and cell mediated immunity. So this is the image that shows the different antigens possessed by this bacteria. So it can secrete exotoxins, vacuolating antigen, CAG toxin, CAG protein, and variety of other antigens, including the enzymes. So prevalence and significance of H. pylori diseases. How far the H. pylori is prevalent? Why it is given so much significance? Why people? Why it is alarming? The diseases caused by H. pylori is alarming. Why? H. pylori mediated chronic gastritis disease is once occurs, it advances as the age advances. So as the age increases, the disease also advances. It is not curing. Whereas in most cases, most of the bacteria, they cause acute infections. At one point of time, by the immunity or by the treatment, they can be, you can get rid of it. But this disease does not easily get rid. It does not easily get off the body. Some epidemiological studies which are made on the prevalence of the disease are based on the serological detection. They're all pertaining to the gastric infection. No, the epidemiological studies help us to find out how much, what is the prevalence, what is the percentage of disease in a particular population. So these epidemiological studies are based on the detection of anti-H. pylori antibodies, especially in people with gastric infection. So not all the people are examined. People who are complaining of gastric infection, they are examined and they are diagnosed for the presence of, for the detection of anti-H. pylori antibodies. So the epidemiological data shows that surprisingly, 50% of the world population are affected by the H. pylori. You know, it is surprising. So who knows? One among us, or our neighbors, our friends, or colleagues might be H. pylori infected. But still, you don't have to worry, because most of these infections, or majority of the H. pylori infections, are subclinical in nature. Subclinical in the sense, there is no symptoms, there is no complications, there is no major pathogenic effect or pathological effect. So, although we harbor this bacteria as a cause of infection, the complications are rare, or complications are not substantial. So overall prevalence, if you see the overall prevalence of H. pylori disease, it is in the range of 40 to 80% in the case of gastric patients with gastritis, and the proportion of this disease is very common in developing countries, poor countries and underdeveloped countries. So this disease does not occur only in children or adolescents or adults, but also in pediatric group. There was a Romanian study, in a study by Sur et al. In Romania, who proposed that, who revealed that approximately 12% of the infants, the neonates, infants, are infected with this bacteria. So it does not spare any group of people, any age group of people. So eradication of this disease caused by H. pylori is a very, very difficult task unless you put in extraordinary effort and use amalgamation of different antibiotics. So you need not only a single antibiotics, multiple antibiotics, multiple treatments are required to eradicate this H. pylori. So what is the etiology and complication of H. pylori disease? What causes the H. pylori? What brings in the complication of H. pylori disease? So primary colonization of H. pylori occurs in the gastric epithelium. So the gastric epithelium has some specific receptors, special receptors that attracts the H. pylori, and so the colonization of H. pylori takes place. And not only the colonization, strong colonization of these bacteria, but also the failure of immune mechanisms to eliminate H. pylori makes this disease a worst disease. So the disease is characterized by prolonged infection. The infection is prolonged for many years. The infection occurs, pronounced inflammation occurs. So often there is inflammation. That is why the people with Helicobacter infection, they often complain of stomach ache or vomiting, nausea, and persistent tissue damage. These are the common clinical manifestations, conditions associated with H. pylori disease. The clinical manifestations occur in 10 to 15% of the infected individuals, and the normal outcomes, the disease outcomes are the peptic ulcer disease, the most common disease, gastric cancer. In some cases, you may have, in most cases, you have chronic gastritis. Gastritis, a reflux of the intestinal or stomach contents. This is a very common condition. Besides, you have peptic ulcer disease, gastric cancer, and in some cases, lymphoma of malt. What is malt? Mucosa-associated lymphoid tissue, which is found along your entire respiratory tract and digestive tract and urinogenital tract. So this lymphoma cancer condition can be caused by this bacteria. The most serious complication. What is the most serious complication of this H. pylori disease? The development of carcinogenesis. Carcinogenesis. Carcinogenesis is a type of cancer, carcinoma. That's the cancer of the epithelial cells. So this bacteria is very, very closely associated with carcinogenesis. Understanding the significance of this, the World Health Organization classified H. pylori as class 1 carcinogen. Carcinogens are usually chemicals, but interestingly, this bacteria is considered or categorized, classified as a class 1 carcinogen. You have different classes of carcinogens. Class 1 carcinogen means the most important carcinogen. So that the infection can, in most cases or in chronic cases, can lead to cancer. So the disease is associated with prolonged inflammatory immune responses, especially in the epithelial cells, which leads to alterations in the cells, mutations in the genes of the cells, so that the cells which have undergone mutation, they undergo altered cell growth and cancer development. So in rare complications, you can find gastric carcinoma in 1% of the cases and lymphoma of malt. So 1% of the cases, infected cases, develop gastric adenocarcinoma. Rarely you can find oropharyngeal carcinoma. So this is how the bacteria infects. It is transmitted through oral route and invades the stomach mucosal lining, where it causes ulceration, not only in the stomach but also in the duodenum, peptic ulcer. So this bacteria is capable of attaching to the epithelial surface of the stomach, and it causes a kind of reaction like drilling the tissues, so that the mucosa, submucosa, everything is eroded. So once invaded strongly, this chronic infection can lead to variety of diseases like malt lymphoma, gastric cancer, gastric ulcer, duodenal ulcer, gastric cancer, gastric atrophy, and acute gastritis, which is very common. So far, we believed that the bacteria is a gastric pathogen, causes only gastric infection or complication in the stomach. But recent research studies says that the bacteria cause complications in various other anatomical sites of the body. So the bacteria has been found in not only in the stomach, but also in some other areas like adenoids, dental plaques, gallbladder, eyes, nose, ears, and skin. So bacteria can be found in other areas too. But primary infection occurs in the stomach, but it can also be found in other areas. So the epidemiological studies that are carried out in with respect to H. pylori infection reveal that it causes an array of infections, array of diseases, which can be immunological diseases, neurological diseases, cardiovascular diseases, hematological, dermatological, hepatobiliary, endocrine, and gynecological diseases. So this bacteria, one bacteria, is capable of causing spectrum of diseases. But one must know that the bacteria is not directly involved in all these complications, all these diseases. So it creates a kind of complication in the stomach, a kind of reaction that affects various other anatomical sites of the body where the diseases develop, the complications develop. So various research studies have been carried out to propose that the bacteria is associated with extra-gastric diseases. Extra means outside the stomach, which can be a metabolic syndrome, non-alcoholic fatty liver tissue, it can affect the liver, coronary artery disease, type 1 diabetes mellitus, a dangerous disease, autoimmune disease, insulin resistance, autoimmune thyroid disease, celiac disease, iron deficiency, and arthritis. So these are all the different references, not limited references, many references are there. You have, which are all proposing that the H. pylori is involved in extra-gastric diseases. So initially, once the bacteria infects the intestinal epithelium, initial symptom would be the gastritis, and chronic infection cause gastric ulcer. In some cases, it can progress to gastric cancer. So that the stomach becomes cancerous. During this gastric ulcer, there occurs extensive immunological reactions, resulting in the production of some immunological inflammatory products, which diffuse into various parts of the body and cause infection of different systems, including neurological, coronary system, and then endocrine system, etc. So extra-gastric diseases, why do they occur? Why do they occur, and how do they occur in H. pylori infection? The reason is that frequent failure of antibiotic therapy. So the antibiotics that are used for H. pylori in the past, in recent days, effective antibiotics are there. So they are the root causes of spread of this infection. So because of failure of antibiotic therapy, the bacteria is capable of surviving for longer period, and it can spread to other anatomical sites. Besides, the H. pylori induces certain immune reactions, and these immune reactions become systemic, means it spread to other parts of the body, so that it affects multiple organs. So what happens? What happens when the infection occurs and the immune reactions are mounted? Initially, during the initial stage of the gastric infection, in the gastric mucosa, some kind of inflammation occurs, so that there occurs infiltration of polymorphonuclear cells. So what are polymorphonuclear cells? The basophils, eosinophils, and the neutrophils. So the neutrophils are the predominant phagocytic cells. So wherever the inflammation occurs, the neutrophils rush to that area and cause phagocytosis. Gradually, when the infection progresses during the chronic stage, there occurs infiltration of mononuclear phagocytic cells. So these polymorphonuclear cells are replaced by mononuclear phagocytic cells like macrophages. Macrophages of different types. So macrophages and monocytes, they infiltrate there and cause further damage. When these cells are present, especially the macrophages are present, they have the characteristic of secreting some pro-inflammatory cytokines. Pro-inflammatory cytokines, what are they? The cytokines are the substances secreted by immune cells that mediate various immunological reactions. When these substances are inducing the inflammation, they are called pro-inflammatory cytokines. So these pro-inflammatory cytokines diffuse from the stomach into various parts of the body, where they induce inflammation of different kinds, which causes complications of different anatomical sites. So this is how the adverse effects are caused in various other anatomical sites. Okay, let us come to the main part of our, the final part of our lecture, that is Helicobacter pylori, a risk factor for autoimmune thyroiditis. What is the connection between Helicobacter pylori and the autoimmune thyroiditis? So, as I just mentioned before, Helicobacter pylori infection causes some abnormal immune reactions. Prolonged chronic infection leads to abnormal immune reactions. In due course, it becomes autoimmune in nature. At one point, some antibodies are produced against gastric cells, gastric antigens. And these antibodies, or these abnormal autoimmune reactions, are considered as risk factor for autoimmune thyroiditis. So the Helicobacter pylori infection causes elevated secretions in the stomach. of some substances called pepsinogen and gastrin. So these substances are usually the substances secreted in optimal level for digestion. But in H. pylori infection, because of the inflammation and stimulation, the pepsinogen and gastrin are secreted in more amount that induces some abnormal immunological reactions. So most common abnormal immunological reactions are autoimmune gastritis related immune reactions. And these autoimmune gastritis further extends, and these antibodies which are reacting with the stomach also react with the thyroid antigens. That is how the autoimmune thyroiditis starts. So you have different experimental proofs that links the H. pylori infection with the thyroiditis. Some scientists explain that there occurs marked reduction of level of T3 and T4 hormones in patients with active H. pylori infection. So this scientist examined that what is the relationship between H. pylori and thyroid. So this scientist found that H. pylori, the persons who are having H. pylori infection produce low amount of T3 and T4 thyroid hormones, which means that H. pylori infection has direct impact on thyroid secretion. Another scientist, Betlot et al. In the year 2004, he found that there occurs a linear regression between anti-H. pylori antibodies and microsomal antibodies. Microsomal antibodies, there occurs reduction of these antibodies after the H. pylori infection is removed. So microsomal autoantibodies are usual antibodies produced in autoimmune thyroiditis. So this person found that there occurs parallel simultaneous increase in antibodies of anti-H. pylori and microsomal autoantibodies. And these antibodies disappear once the infection is eradicated. So some other people who did research on this area found that the Hashimoto's thyroiditis is associated with higher prevalence of anti-H. pylori IgG antibodies. And the Korani et al. In the year 2016 found that H. pylori antigens are found unusually in patients with Hashimoto's thyroiditis. H. pylori antigen, CAG antigen, I told you, the cytotoxic antigen A, which is unusually found in patients with Hashimoto's thyroiditis. Usually, Hashimoto's thyroiditis patients does not show symptoms of H. pylori, but when examined, this scientist found that the patients with Hashimoto's thyroiditis showed the presence of H. pylori antigen in the stool roughly about 40% to 75%. Okay, let us see about the mechanisms by which the H. pylori induces thyroiditis. How does it induce the thyroiditis? The various mechanisms are proposed. About 6 mechanisms are very important. Let us see these mechanisms one by one. Molecular mimicry. As I mentioned before in the beginning of this lecture, mimicry is nothing but the resemblance, close resemblance. So molecular mimicry is one of the mechanisms proposed for autoimmune reactions. Some of the epidemiology and serology based studies confirm that some bacteria and viruses, when they infect, their structure mimic the structures of thyroid cell membranes, so that the antibodies produced against the bacteria and viruses also react with thyroid cell membrane, so that autoimmune disease occur. So this is evident not only with the H. pylori infection, but also some bacteria like Yersinia enterocolytica, most common cause of the enteric disease. So this bacteria is associated with another thyroid disease called Graves' disease. So same way, the infection by H. pylori in the stomach by H. pylori, there occurs the production of antibodies which also react with the thyroid antigens. It has been experimentally proved. So researchers also have established that the H. pylori infection is also associated with Graves' disease, which is Graves' disease usually hyperthyroidism case. So here, over secretion of hyperthyroid, the thyroid also can be caused by H. pylori. So this is how it occurs. So the infectious agent, when it enters, it is the antigens are processed and presented to T cells, and T cells recognize them and stimulate the B cells to produce antibodies. But incidentally, these antigens resemble the structures of some of the structures of our body, mimic the structures of our body, so that the autoimmune reactions are mounted. The second phenomena, or second immune mechanism, is the inflammatory immune response. Production of pro-inflammatory cytokines in the stomach. So the chronic inflammation, chronic infection leads to inflammation. Pro-inflammatory cytokines are produced because of the infiltration of polymorphonuclear cells, as I mentioned before, the neutrophils, the basophils and eosinophils. So these polymorphonuclear cells are slowly replaced under the influence of CD4 cells. You know, the CD4 cells are T cells. T cells are of two types, Th1 cells and Th2 cells, T helper cells. T helper cells 1 are responsible for cell-mediated immunity, and Th2 cells are responsible for humoral mediated immunity. They control them. So these Th1 cells, once involved, they activate the macrophages, so that there occurs the production, the infiltration of macrophages, which in turn secrete some inflammatory cytokines. These inflammatory cytokines, once produced, the inflammation, tissue damage increases. Not only in the stomach, but also these cytokines diffuse out of the gastric mucosa and reach far tissues. So the inflammation which is activated in the thyroid gland results in the autoimmune reaction against thyroid antigenic profile. So the inflammatory cytokines produced in the stomach due to H. pylori infection reach the thyroid gland and cause autoimmunity in thyroid gland. So there is a diagnostic evidence that there occurs heightened chemokine response in patients with autoimmune thyroiditis. If we observe people with Hashimoto's thyroiditis or autoimmune thyroiditis, you can find more of chemokines in the serum. So this is how it occurs. So when H. pylori infection occurs in the gastric epithelial cells, this is the mucosal cell lining, there occurs tissue damage resulting in the production of chemokines. You know, chemokines are the attractants, chemoattractants that attract polymorphonuclear cells. So they are recruited in the area which engulfs the damaged tissues, and further the macrophages, mononuclear phagocytic cells, are recruited there under the influence of the Th1 cells, which further secrete interleukin 2 and interferon gamma. So these interferon gamma, interleukin 1b, beta, demon necrosis factor alpha, these are all, interleukin 8, these are all together collectively known as the pro-inflammatory cytokines. So these cytokines will further aggravate the reaction and recruit more and more cells, causing more damage. So this is how the gastric ulceration occurs. So the ulcer, the tissue damage, is because of the inflammation. The next mechanism proposed is the antibody cross reaction. So the autoimmunity, what is developed in the gastric mucosa, can eventually lead to autoimmune disease. As I mentioned before, you know, in the gastric mucosa, there are some cells called parietal cells. You would have heard about them. Parietal cells, they are very important cells in the stomach. These cells, structurally, they have some components, some chemical components that resembles the thyroid peroxidase enzyme. You know, thyroid peroxidase enzyme is a very important enzyme responsible for thyroid secretion, thyroid production and secretion. So interestingly, this parietal cell and the thyroid peroxidase, because they have structural similarity, because of the possession of some peptides. Some peptides are particularly homologous 11 residue peptide. There is a peptide which has 11 residues, amino acid residues. And these two, this is present in both these cells. So the antibodies produced in the stomach against the parietal cells due to H. pylori infection also react with the thyroid peroxidase. So that what happens? In due course, the thyroid peroxidase function is affected, resulting in hypothyroidism. In addition, the CAG antigen I told you, the cytolytic antigen A of the H. pylori, it is coded by a group of genes called pathogenicity island, CAG pathogenicity island in the bacteria. The bacteria possesses a group of genes, CAG pathogenicity island. They comprise of some nucleotide sequences. Interestingly, they are similar to the genes coding for endogenous peroxidase. See the relationship, how it is linked. The bacteria possesses a group of genes, the CAG pathogenicity island, and these genes possess some nucleotide sequences which are closely related or similar to the genes coding for endogenous peroxidase, which is associated with the thyroid gland. So that what happens? This antibody produced against the CAG antigen can also react with the follicular cells of thyroid gland. So this is how it happens. So resemblance not only with the structure of parietal cells and the thyroid cells, but also resemblance occurs between the CAG antigen of H. pylori and the endogenous peroxidase of thyroid gland. So cross reaction between these two results in autoimmunity. So cross reaction is, what is cross reaction? An antibody produced against one antigen also reacts with another. And the antibodies are usually specific. They react with only one antigen. They do not react with another antigen. But when the antibody reacts with two antigens of structural similarity, two antigens having structural similarity, this is called cross reaction. So cross reaction, antibody cross reaction is the basis of autoimmune thyroiditis, which is induced by H. pylori. So it is also experimentally proved that there occurs a close association of levels of immunoglobulin G antibodies produced against CAG antigen and the antibodies to thyroid peroxidase. So the antibodies of produced against CAG antigen and thyroid peroxidase, they have a close resemblance, or they are more or less same. Then comes another important component, a mechanism called dysregulation of vitamin D receptor. What is it? Nowadays we talk more about vitamin D. So vitamin D is a very, very essential vitamin which we synthesize in our body under the triggering factor of sunlight. People say if you are exposed to sunlight, you get vitamin D. So it is not that sunlight provides you vitamin D. Sunlight induces some mechanisms that helps in the synthesis of vitamin D in your body. How does it occur? You know, there is a component called 25D. This 25D is a compound, a chemical compound present in the subepithelial cells of our body, below the skin of our body. We have some compounds called 25D compound. This compound is activated. This compound is activated under the influence of sunlight, so that an enzyme called 1-alpha hydroxylase, there is an enzyme called 1-alpha hydroxylase that causes conversion or activation of this 25D into 125D. So that what happens? This 125D is the one that causes kind of stimulation in the chromosome, in the genes present in the cells, so that the vitamin D is transcribed. Vitamin D gene is transcribed and the vitamin D is produced. This is how the mechanism of vitamin D is occurring. But this the compound 125D, if it is not utilized properly, and if it is not received by the thyroid receptor, if it is not received by the vitamin D receptor, it will be disoriented, because this compound 125D has affinity for both vitamin D receptor as well as the thyroid receptor. If the vitamin D receptor is impaired, the compound 125D will get attached to the thyroid receptor, alpha thyroid receptor, resulting in dysregulation of the transcription of thyroid production. So what happens is, the H. pylori infection has been found to interfere with the vitamin D receptor. So vitamin D receptor is interrupted because of the H. pylori infection. So that what happens? vitamin D receptor cannot receive 125 compound. I told you the compound 125D. So also I told you compound 125D has attraction for the thyroid receptor. So it has attraction for both. If vitamin D receptor is not available, the compound 125D will get attracted to thyroid receptor, so that it will cause impairment of thyroid. So this is another mechanism by which the H. pylori infection indirectly cause autoimmune thyroiditis. Then comes the insulin resistance. The type 2 diabetes mellitus. In this case, the anatomical and physiological anomalies of thyroid gland can be caused by insulin resistance. So the thyroid gene expression, if you see, thyroid is because of the expression of some genes in the thyroid gland, which are influenced by some signal pathway. So the expression of thyroid gene needs some signal pathway which is concerned with insulin-like growth factor. There is a growth factor called insulin-like growth factor. This growth factor is essential, which is associated with the insulin for the expression of insulin gene. So experiments proved that H. pylori infection causes alteration in insulin pathway. So that the insulin pathway, because it is affected, the signal pathway is not available for thyroid gene express, so that the thyroid gene expression is affected, so thyroid dysfunction occurs. So some scientists have proved, like Junik et al. In the year 2006 proved that there occurred negative linear correlation between thyroid volume and concentration of thyroid stimulating hormone in type 2 diabetes mellitus. So insulin pathway, signaling pathway is essential for thyroid gene expression, which is affected by H. pylori, so that the thyroid disinfection starts. Then comes the alteration of HLA. Alteration of HLA. What is alteration? How does the alteration occur? HLA, you should know what is HLA. Human leukocytic antigen. This is self-antigen. So when I in the beginning of this lecture, I said about self-antigen and foreign antigen. The self-antigens of our body are referred in terms of HLA, human leukocytic antigen, which are coded by a set of genes. So the genes, the HLA genes are found in all over the body, and every cell has its own HLA antigen. Some HLA antigens are responsible for some immune functions, some are for adverse functions. So it was found by some research study that the H. pylori infection causes alteration of HLA self-antigen. So the HLA antigen is altered in such a way so that the autoimmune disease is developed. For example, as long as your body structure or your antigens of your body remain intact, as such, immune system recognizes it as self-antigen. Any change in the structure in your body due to infection or any damage, physical damage, you may get autoimmune disease. For example, there is a kind of disease called autoimmune disease due to sequestered antigens. Sequestered means the antigens which are hidden. For example, your lens protein, your eyes lens protein. So the lens protein, the eyes, they do not have vascular connection. It is enclosed, it is encompassed, enclosed. When any damage to eye occurs, the lens protein is exposed. The lens protein is usually, it is in a closed compartment. That is why it is called sequestered antigen. When this, as long as it is inside, it is a self-antigen. When it is damaged and exposed, it is considered by mistake by a foreign antigen, so that autoimmunity is developed. So it is the genes coding for HLA that the alteration of which causes autoimmunity. So there is a gene called HLA DRB1 0301. It is a very, very important marker for autoimmunity. This gene is usually remaining in a suppressed form, but in the case of autoimmune persons, the persons with autoimmunity, this gene is more expressed. So this allele usually is involved in prompting cell-mediated immunity or humoral-mediated immunity against self-antigens, self-structures, autoimmunity. So the alteration of this allele is caused by H. pylori infection, by some molecular mechanisms. So this alteration of gene result in development of autoimmunity. The frequent target of this autoimmunity mediated by alteration of this gene is thymocytes, thyrocytes. Thyrocytes are the cells of the thymus. So the last mechanism is the epiphenomenal immune mechanism. So epiphenomenal immune mechanism is a immune mechanism which is progressive immune mechanism. Immune mechanism taking place in one place, progresses to another place, and sometimes it becomes systemic. Systemic means all over the body. So you have, as I mentioned before, there occurs a molecular mimicry, similarity of the structure of antigens of H. pylori and others. So the antigens, CAG antigens or other antigens of H. pylori, they mimic the enzyme of parietal cells. You know, the stomach cells produce various enzymes. Some of the important enzymes are H plus, K plus, ATPase. These are very important enzymes for mediating metabolic activities in the stomach. So it so happens, it is recently found that the antigens of H. pylori mimic, they have structural similarity with the enzyme produced by parietal cells. So that triggers the immune mechanisms, immune reactions. So the dendritic macrophages, you know, the macrophages are of different types, dendritic, alveolar, Kupffer, the microglial, different macrophages are there. So the dendritic macrophages are so important. Why? Because they are functioning as antigen presenting cells, APC, antigen presenting cells. So they present, what is the role of them? You know, if you compare the T cells and B cells, the T cells recognize the foreign antigen only if it is presented by an antigen presenting cell. So it has to be, the antigen, foreign antigen has to be consumed, broken into pieces and presented to T helper cells. So the dendritic macrophages, they present the bacterial epitopes to naive T cells. Naive means unstimulated T cells. So they produce, once the naive cells are activated, they produce clones of autoreactive Th1 cells. T helper cell 1 cell. Th1 means T helper cell 1 cells. So what happens once the T helper cell Th1 cells are activated, they secrete a series of lymphokines, which causes recruitment and infiltration of cytotoxic T lymphocytes, another type of lymphocytes which are responsible for tissue damage. You know, you have T cells like T helper cells, cytotoxic T lymphocytes, suppressor T lymphocytes, delayed hypersensitivity T lymphocytes, etc. The cytotoxic T lymphocytes are very important in mediating cell-mediated immunity. What is the role of them? Their role is to destroy the cells, our body cells. What is this? Why cells have to damage the immune cells have to damage our cells? No, they don't damage the normal cells. They damage and destroy the cells that are infected, that have become dysfunctional, which are not working at all. Only such cells are recognized and damaged, including cancer cells, virus-infected cells, or transplanted cells. So the macrophages, what do? The dendritic macrophages collect the antigens of H. pylori and present them to T helper cells, T helper cell 1, Th1 cells, which trigger the production activation of cytotoxic T lymphocytes. So the cytotoxic T lymphocytes, as I mentioned before, they are responsible for cell damage. In technical term, we call it as apoptosis, cell damage, cell death. So this apoptosis is induced by some kind of lymphokines secreted by cytotoxic lymphocytes, including interferon gamma, tumor necrosis factor alpha, etc. So that there occurs parenchymal tissue destruction in the gastric mucosa. So that the destruction of the gastric mucosa results in extensive immunological reactions, and there occurs epiphenomenal production. Epiphenomenal reaction means extended immunological reaction resulting in the production of antibodies to the self-antigens. So due to vigorous immunological reaction, antibodies are produced both against the bacteria as well as the stomach cells, the gastric cells, so that the parietal cells are damaged and the antibodies produced against these antigens also react with the thyroid antigens like thyroperoxidase, thyroglobulin, and other antigens, so that the immunological reaction extends, resulting in dysfunction of the thyroid gland. So this is how the epiphenomenal immune reaction occurs. So Helicobacter pylori infection in the gastric mucosa epithelial cells causes immunological mechanisms, so that antigen presenting cells present the antigen to the Th1 cells, resulting in the production of cytotoxic T lymphocytes that further damages the cells through production of some apoptosis inducing substances, including perforins and other substances. So the immunologically, the fast reaction is important because this is important for self-destruction, death receptor. This is called death receptor. So extensive damage is because of epiphenomenal immune reaction. So we have come to the last part of your my talk, that is the conclusions. So I have given you three important components in my lecture. The first thing about the biochemistry, that is thyroiditis, description, its prevalence, then Helicobacter pylori, the diseases, complications, and extragastric diseases, and the link, the third component, link. I explained to you various experimental proofs and the mechanisms. So what is the conclusion? What is the take-home message out of this lecture? So autoimmune thyroiditis or Hashimoto's thyroiditis is grabbing much of attention in recent days because of its global spread. It is widespread nowadays because it causes severe morbidity. Morbidity means damage or malfunctioning of organs. So infections caused by H. pylori is considered very serious because it is chronic. In most cases, it is chronic. The clinical manifestations can be sometimes subclinical, or sometimes may cause minor morbidity or sometimes mortality, because it is involved in cancer, gastric cancer, peptic cancer, and other types of cancers, induced cancers. So the important point is that the H. pylori can cause extragastric complications. The infection leads to immunological reactions which diffuses into other parts of the body, leading to extragastric complications. This is mainly because of the ability of the pathogen to overcome the immune mechanisms and other therapeutic regimes. So it is neither responding to the treatment nor the immunological mechanism, so that it sustains for longer period and cause damage to the stomach as well as other organs. So there has been a series of research studies in the past 2 decades which attribute the link between the H. pylori infection with the pathogenesis of autoimmune thyroiditis. So the chronic gastric infection, very important. People who have gastric infection, they have to be careful in treating it so that they can avoid the further immune and molecular mechanisms that can lead to the production of autoimmune thyroiditis. So these are all some of the references I utilized for preparing my article and this lecture. So I thank you one and all for this opportunity to share my information with every one of you. I'm prepared, I'm open here to answer your questions if you have anything.
Thank you, sir, for giving us an insight into this pathogenesis. The session was an eye-opener to all, sir. Your lecture was very crystal clear and very much informative about the Helicobacter pylori, sir. There are a few questions from the participants.
Yes.
Some of the questions. Is there any vaccine for this Helicobacter pylori?
Yes, vaccine is available, but it is not promising to be 100% successful. As usual, the vaccine, the bacteria is capable of changing its antigenic structure frequently. So the periodical vaccine is advisable. So wherever the prevalence is common, periodically it can be given, just as in the case of influenza, and other common epidemic diseases.
Then second question, is there any early method to detect this infection?
Yes, methods are there. As soon as you experience any kind of symptoms, gastric symptoms, or any kind of abnormality in your digestive tract, you can go for some serological tests. As early as the infection is onset, you can find the detection of IgM antibodies to H. pylori. And also some biochemical tests like urease test is available, which can be done in any laboratory as soon as we experience any kind of symptom.
Then third one, what food we should avoid when we have this bacteria, Helicobacter pylori?
Important point is that the hygiene. So hygiene matters. For any kind of infection, say it is a bacteria or virus or fungus or parasite, hygiene matters. As we know, this bacteria causes the infection of digestive tract, particularly the stomach, and the transmission is through oral, fecal-oral route. Personal hygiene is very important. So just like nowadays we practice hand wash and other things nowadays and keep the place clean and sanitation. So the personal hygiene and sanitation, especially with respect to the food and water, is very important. So ensure that the food is properly cooked and it is properly served, and the person handling the food, serving the food, eating the food, maintain the hygiene, and food is not left to stale, and spoiled food should be avoided. Especially the water contamination is the main cause of spread of this infection. So when you use the water for not only drinking but also for cooking or even for washing the utensils, the water you should ensure that the water is of good quality. It is not contaminated by any means. Yes.
Rita ma'am, unmute and speak.
Question sessions over, sir. I'll deliver a vote of thanks now. The roots of all goodness lie in the soil of gratitude and appreciation. On behalf of our Department of Science, Anai Violet Arts and Science College, from the depth of our hearts, we thank our invited speaker, Dr. Tyagarajan, Assistant Professor and Coordinator, Northern Border University, Kingdom of Saudi Arabia, who have readily accepted our invitation with no second thought in spite of his busy schedule, and enlightened the session with his thoughtful lecture. We feel the purpose of the webinar has been accomplished because of your contribution in your specialized areas. Once again, thank you all very much. Okay, a gentle reminder, our next session starts by 4 o'clock.